Hi everyone, today I'm going to explain to you the central dogma of molecular biology. The central dogma of molecular biology is an explanation of the flow of genetic material within a biological system. It is a process that makes DNA, which goes to RNA, and RNA makes a protein. But today, I'm going to talk to you about what happens between the arrows. The first step is DNA from DNA. This is where replication occurs. The main enzyme that drives this process is DNA polymerase. The second step in this process is from DNA to RNA, and this is when transcription takes place. The main enzyme that drives this process is RNA polymerase. And the last step in this process is from RNA to protein. During this time, this is when translation occurs. This process of translation is due to 50 S ribosomes binding to the codons. Now, DNA replication occurs once in the lifetime of a cell. The purpose is to serve as hereditary material to make for progeny. The process is the manufacture of an exact copy of DNA contained within a cell. Now, in this slide, I'm going to show you the process of initiation. DNA replication begins at the origin of replication. This is a specific point on the DNA molecule where replication can begin. The enzyme helicase attaches to the DNA molecule. It catalyzes the opening of two strands of DNA, which lead to the breakage of hydrogen bonds between the base pairs. This enzyme then attaches to the two strands of DNA when the single-stranded binding proteins attach to the section of the DNA molecule. These proteins hold it open so DNA doesn't close and hydrogen bond. Now, the next step is elongation and termination. The enzyme DNA polymerase starts this process of replication. The enzyme only has the ability to add new DNA nucleotides to an already made chain of nucleotides. This is why the enzyme primase, which is an enzyme that assembles RNA primer at the origin of replication. This allows the DNA polymerase to now attach to the strand. Now the next step can happen. This now allows the DNA polymerase to add nucleotides to the RNA primer and begin the construction of a complementary strand. After this, the RNA primer is removed by a nucleotide replacement enzyme and then replaces with the correct nucleotide sequence. Now, DNA polymerase only has the ability to allow replication to happen in one direction. This is why there are two ways to copy strands of DNA. The DNA molecule runs anti-parallel. One strand is replicated completely, while the other is done in Okazaki fragments. These Okazaki fragments need to separate the RNA primers in order to function. So the enzyme primase attaches to the lagging strand so DNA polymerase can start replication on this strand. When the Okazaki fragments are synthesized, the RNA primers are then replaced with the right DNA nucleotide by the nucleotide replacement enzyme. This process allows these Okazaki fragments to be bonded together to make a continuous complementary strand. Now, the next step in this process is transcription. In this slide, I am going to show you initiation. Transcription is a process of producing an RNA message from DNA. The process begins when the RNA polymerase binds to a subunit of the mRNA called the sigma factor. This sigma factor helps the enzyme recognize the promoter. RNA polymerase binds the DNA and separates it into two strands. It adds nucleotides to the 3' end of the growing RNA molecule. The promoter consists of the initiation site where transcription begins. After RNA has bound to it, the sigma factor leaves and elongation can now finally occur. Now, in this slide, I'm going to show you elongation of the molecule. Now, RNA polymerase moves along with DNA and performs two functions. It first untwists the template of the DNA at about 1 turn, 10 bases at a time. Then it adds nucleotides to the 3' end of the growing mRNA molecule. As RNA polymerase moves along the DNA, it creates a new stretch of DNA. Now, if you see on the bottom slide, this is where termination occurs. The process finishes when RNA polymerase reaches a termination site on the DNA. The most common stop sequence is AATAAA. Once here at the termination site, a hair loop structure forms in the RNA, actually forms in the DNA, causing the RNA polymerase to separate from the DNA. When done, no more RNA nucleotides are added, and the mRNA is released. Okay, the next process in the system is called translation. Translation is the process of building a polypeptide. In this slide, I am showing you initiation. Translation initiated by the forming of an initiation complex. This complex is made up of a 30S ribosomal unit, FMET, with its formal methynyl, tRNA, and messenger RNA. Then, the 50S ribosomal subunit joins the complex. This joining allows the 70S ribosomal subunits to now bind to make a polypeptide chain. Ribosomes have 3 binding sites. 1 to attach to mRNA and 2 sites for attaching tRNAs. The P site is the peptidyl. It holds the tRNA and attaches to the growing polypeptide chain. The A site is acceptor. It holds the tRNA attached to the next amino acid to be added to the chain. And the last site is the E site. It is the exit site. It is where transfer RNA are released. When FMET binds to the P site, tRNAs has the ability to recognize the next codon and then carries the amino acids to the A site. The next step in this process is called elongation. FMET is carried by transfer RNA to the P site and then joins the amino acids by tRNA that ended the A site. The ribosome moves a distance of 1 codon, and the tRNA that carried the FMET is then released to the E site. After this has occurred, the tRNA that carried the next amino acid moves into the A site, where the anticodon of the tRNA matches the codons of the mRNA. The ribosomes just keep moving a distance of 1 codon each time. As this shift occurred, the 2 amino acids of the tRNA in the P site are moved to the new amino acid, and the second transfer of the tRNA is finally released. The ribosomes keep moving along with mRNA as more and more amino acids are added to the growing polypeptide chain. Now, the last step in this process is called termination. This is when the process of elongation is stopped when the polypeptide hits a stop codon into the A site. A stop codon doesn't have a corresponding tRNA. This allows the ribosomes, the mRNA, and the proteins to be released. Now the process is finished, you have a protein. Now, a new protein has been produced by this cycle, and it may have to be further modified to be functional. Now, this protein can be used by addition of molecules of sugars, lipids, or phosphate groups. A single chain may be cleaved off into two pieces, or multiple polypeptides may be joined together to make a functional protein. An example of this is hemoglobin. Hemoglobin is made up of 4 polypeptide chains. So thank you all for listening to my video, and I hope you all learned a lot from this. Have a good day.